In this process, we begin by taking a blood sample and placing it into the AccuCyte separation tube. You can see the black float at the bottom of the tube. The tube is spun in a centrifuge, and the plasma separates at the top, the buffy coat in the middle, and the red cells at the bottom. We then use a cyte seal ring to clamp the tube so that the red cells are separated from the rest of the sample. This is done using an instrument called the cyte sealer. The separator tube is inserted into the cyte sealer, its height is adjusted, the lid is closed, and then the instrument clamps the brass ring around the outside of the tube, effectively walling off all the red cells beneath. So now we see plasma, buffy coat layer, and then we see the sealing ring and the red cells beneath. The plasma is aspirated and saved for cell-free analysis. And then an inert water-soluble displacement fluid is added to the tube. This displacement has a greater density than all the cells that are above the sealing ring. The collector is inserted into the separator tube. Excess fluid that comes out of the needle pointing upwards is removed. The isolation is placed into isolated collector, and now when this is spun, the heavy density fluid dislodges the cells from around the float all the way through the collector, and now we have them in a very small volume in an Effendorf tube. Next, we prepare slides from the buffy coat sample. We mix the cells with an adhesion liquid that has been designed to allow the cells to form an adhered monolayer on a microscope slide. A slide spreader device that RareCyte has created is used for this purpose. We place a portion of the mixture onto the microscope slide and simply pull the blade right across the slide. This produces a monolayer smear. It's then dried, and it can be stored for a long period of time, and this is what goes onto the automated immunostainer for staining.
After the slides are stained, they're placed into the CyteFinder automated scanning instrument. The CyteFinder has locations for 2 slides, or for a slide and a tube holder. After scanning the slide, images are presented to the reviewer, such as these images, and individual channels may be used to identify the characteristics of the cells for confirmation as circulating tumor cells. High-resolution imaging allows for even greater detail at 10x or 40x. Next, a cell of interest can be picked off the slide using the cyte picker. The system centers the cell by a mouse click. The needle is lowered, and then with a single click, a routine is begun that allows the dislodging of the cell off the slide and then sucking it into the needle tip. You'll see the cells on the outside move, whereas the cell in the bore of the needle remains stationary. Now what you'll see in a moment is that the cell in the middle disappears as it is being sucked into the needle tip. It's no longer present on the slide. Now the needle is raised from the slide, the stage is moved so that a tube is underneath the needle. The needle is placed into the tube. Fluid is expelled through the needle into the tube, and because the tube has a flat bottom, we may image the cell that has come through the tube. As you can see in the upper left, there is a cytokeratin positive cell that has been expelled from the needle tip. Thus, we have completed a chain of custody of identification of the cell, picking, and then visualizing the cell once again in the tube.